Hello friends, I am Dr. Gaurav Bharadwaj, Assistant Professor in the Department of Biotechnology. I will discuss very basic and very important topic for research purposes now. So this topic is the plasmid. Plasmid. First of all, before the starting plasmid, we have to discuss some points regarding prokaryotic cell and eukaryotic cell. What is the basic difference between eukaryotic cell and prokaryotic cell? Prokaryotic cell having nucleus undeveloped form, yeah, there is no nucleus. Of eukaryotic cell have well developed nucleus. This is the one basic difference. There are so many differences between the prokaryotic cell and eukaryotic cell, but we talk about the plasmid. So plasmid is found inside the prokaryotic cell. So now we talk about only prokaryotic cell. So plasmid is found inside the prokaryotic cell. So we discuss this plasmid in the some basic points. What is plasmid? Types of plasmid. And most important point, what is the role of plasmid in current research era? Basically, most of the students knows what is plasmid, types of plasmid, but don't know about this point. So we starting from first point. So as we discussed, what is plasmid? Plasmid found inside the prokaryotic cell. Second, plasmid is the extra chromosomal DNA. Why extra chromosomal called it? Because it is found extra from the main nucleus. ये मेन न्यूक्लियस के एक्स्ट्रा पाया जाता है, इसलिए उसको हम बोल रहे हैं एक्स्ट्रा क्रोमोसोम. It provide the extra features to the prokaryotic cell or bacterial cell because it having the unique type of genes. Basically, most of the genes found inside the nucleus, you know well, okay, but some extra genes that provide the extra quality to the cell, that is chromosomes, extra chromosomal DNA. So it is also known as plasmid, also known as extra chromosomal DNA because it has the extra genes that provide the extra features to the cell. So first point that is found inside the prokaryotic cell. That is second is it is the extra chromosomal DNA types. It is of three types. First, what is first? R plasmid. That is it. If plasmid have the genes for resistance, that is R plasmid. Many antibiotics does not affect the body. Why? Because bacteria या prokaryotic cell become the resistance against that particular antibiotic. Why? Because this plasmid found inside the R plasmid found inside the bacteria. Second is the F plasmid. F plasmid, that is fertilization plasmid. Fertilization plasmid help in the fertilization of the bacteria. Third one is most important, cool plasmid. That is colicin. Some strong bacteria releases bacteriocin strongest chemicals that inhibit or kill the lowermost of the bacteria. So they grow its family around us. Okay. So next, what is the role of in the research? Because this plasmid having the multiplicative capability, that is it form the multiple copies inside the cell. Multiple copies inside the cell. So this feature we use in the research. Okay, if we take one gene and incorporate in the plasmid, this is the plasmid, incorporate our desired gene. If this plasmid we drop out inside the bacterial cell, it multiply and form multiple copies. So this also act as a multiplicative, replicative copies. So it is this role of the plasmid we uses in our research purpose. So it is the natural means of the multiple copies and PCR is the आर्टिफिशियल मीन ऑफ द कॉपीज। इन दोनों ही मेथड से हम कॉपीज बना सकते हैं, मल्टीपल कॉपीज बना सकते हैं अपनी डिजायर्ड जीन्स का, बट दिस विद द हेल्प ऑफ प्लाज्मिड इट इज द नेचुरल मेथड और विद द हेल्प ऑफ द पीसीआर पॉलीमरेज चेन रिएक्शन इट इज द आर्टिफिशियल मीन ऑफ द replication. So plasmid की main most important feature, what is the plasmid is found in the prokaryotic cell. Second, it is the extra chromosomal DNA because it provide the extra features to the prokaryotic or eukaryotic cell, or it is extra chromosomal DNA or role it having the capability to multiplicate. So this feature we uses in our research. Desired gene we incorporate in the plasmid, and this plasmid if incorporate in the living bacterial cell it multiplicate. So it multiplicate, our desired gene also multiplicate. So we get at the end of experiment a large number of genes as well as plasmids. So this is the main role of the plasmid in our research purposes. So further we added new videos also which are very toughest topic, and students suffer a lots of problems. So watch my videos and subscribe it.